Hello beautiful people, welcome to my YouTube channel Successful Learning. So in this video we will discuss a very important topic of a bioinformatics, that is a BLAST. So what is a bioinformatics? Bioinformatics is nothing but it is a branch of a biotechnology which deals with the study of a biological aspect of a life using a using a information technology. Okay, so BLAST is nothing but BLAST stands for, basically what is a BLAST? BLAST stands for a basic local alignment search tool in an algorithm. It is a algorithm or a program for comparing a primary amino acid sequence or a biological sequences, primary biological sequences such as an amino acid sequence or a different protein or a nucleotide. Okay, a BLAST search enables a researcher to compare a query sequence with a library or a database of a sequence. So through a BLAST we can, or a generally researcher can compare the query sequence and a database sequence. Okay, so what is the similarity between, what is the similarity in their sequences, what is the similarity in their structure, whether they belong to a same species, so etc. Through this we can do or research for a or a compare a query sequences with a library database of a sequence and identify a library sequence that resembles a query sequence. Basically BLAST is a tool from which we can compare, okay, we can compare a query sequence, query sequence that are similar to that query sequence which we are searching for. Okay, so now BLAST is a program that can either be downloaded. So BLAST is a program or a computer program that can be downloaded or run on a command line utility program that is called as a BLASTall. Okay, BLASTall, or accessed for a free over the web. So it is here we can access free over the web. So BLAST web server. So this BLAST web server which is hosted by NCBI. So what is a NCBI? NCBI is a National Center for Biotechnology Information. Okay, so NCBI. So basically this BLAST web server which is hosted by NCBI allows anyone with a web browser to perform a similarity searches. So basically BLAST we can BLAST program can be hosted by NCBI. This allows anyone which has a availability of a web server. Okay, that's it. We can compare or perform a similarity searches against a constantly updated databases, because in a NCBI the databases are constantly updated. Okay, so new databases, recently updated databases are there, so we can compare with those updated databases of a protein, either a protein sequence or a DNA sequence. Okay, now next, what is a BLAST? And BLAST is based on a program, is based on a open source format, giving everyone access. So this has led to a creation of a several BLAST spin-off. So there are handful of different BLAST program available which can be used. So there are many different types of a BLAST program. So basically we call it as a BLAST family. There are many different types of a programs which are different types of a program available, through which we can attempt, or we can compare a query sequence against a database sequence. So these programs and their details are listed below. Here you can see. So as I told you, BLAST is actually a family of a program. It is not an individual program. In this individual program, it has a many programs for which are included. Okay, so first one by one we will go through this. First one is a nucleotide BLAST, or we can generally call as a BLAST-EN. So this program gives a DNA query, returns the most similar sequence from a DNA database. So here it is nothing but nucleotide BLAST or a BLAST-EN. In this program we just search for a DNA sequence against a DNA sequence database. So the sequence which we are searching, this sequence is called as a query sequence, and okay, that's called query sequence. So in a BLAST or a BLAST-EN, in this program we just search for a DNA sequence with those of a DNA sequence database. Okay, next is a second one is a protein BLAST, that is a BLAST-P. Here P represent protein and their N represent nucleotide. So what is BLAST-P? So in this program it is similar to nucleotide BLAST, because in a nucleotide BLAST or a BLAST-N we compare a nucleotide sequence with a nucleotide query sequence with a nucleotide database sequence, but here we compare a program or a compare the program with a protein query sequence. Similar, we are searching for a most similar protein sequence in a database. So third one is a position specific iterative blast, PSI blast. Okay, so this program is used to find a very distinct relative of a protein. First, a list of all closely related protein is created. Okay, so this is a program. So position specific iterative program. So this program is used to find a distinct relative of a protein. First, a list of all closely related protein is created. This profile and these proteins are combined into a general profile sequence, which summarizes the significant features in these sequences. Okay, and a query against a protein database is then run using this profile, and a large group of a protein is found. So this large group is used to construct another profile and process is repeated. So basically it is nothing but, in this program we used to find a distinct relatives of a protein. Okay, first we form a list of all closely related protein, so whether they are sequence are closely related, the domains. Okay, so first we form a list of all closely related proteins, and then this combined profile is then we use this query against this protein sequence. So this list or a profile we use as a query sequence, and we query this against a protein sequence database, and this process is continued. Okay, so by including related proteins in this search, PSI BLAST is much more sensitive in picking up distinct evolutionary relationship than a standard protein BLAST. Okay, so because it forms a profile of very closely related proteins, so due to this it is much more sensitive in picking up a distinct evolutionary relationship. Okay, then that of a standard protein BLAST, that is a BLAST P, right? Okay, so in next third one is a fourth, basically it is fourth nucleotide 6' translation protein. So what is nucleotide 6' translation protein? So this program compare the 6' conceptually translation product of all the nucleotide query sequence, both stand against a protein sequence database. So nucleotide 6' translation protein BLAST, this is called as a BLAST X, and in this program it is nothing but, in this program we compare all the 6 frames or 6 dimensions of a translated product of a nucleotide. Okay, we search for we compare all the 6 frames or 6 dimensions of a translated nucleotide query sequence of both the strands against a protein sequence database. So as you know, in a nucleotide BLAST, protein BLAST, we compare a nucleotide sequence with a nucleotide database, protein sequence in a protein database, but here we compare the translated nucleotide sequence, all the 6 frames are dimensions are compared against a protein sequence database. Now fifth one is a nucleotide 6' translation, T BLAST X. So this program is the slowest of all the BLAST family. Okay, because so reason for this slow is that it translates a query sequence, right? The sequence which we are searching is called as a query sequence. So now in this program, or a nucleotide 6' translation program, in this program we query the nucleotide sequence in all the 6 dimension frame and compare it against all the 6 frame translation of nucleotide sequence database. So we are searching all the 6 frame nucleotide sequence against all the 6 frame of a protein, all the 6 frame of a nucleotide sequence database. So that this is the reason this is a very slowest of all the BLAST family. And the last one is a protein nucleotide 6' translation, T BLAST N. So this program compares a protein query against the 6 reading frames of nucleotide sequence. Basically this is nothing but, in this program we compare a protein sequence against all the 6 dimensions or a 6 frames of a nucleotide sequence database. Okay, right? So next one is the what are the uses of a BLAST. Okay, so there are many uses of a BLAST. So BLAST can be used for a several purpose, right? So these include identifying a particular species, locating domains, establishing phylogeny, DNA mapping and comparison. So first use is identifying species. So with the use of a BLAST, we can correctly identify a species or find a homologous species. As you know, when we are comparing a protein sequence or any other sequences against a database, so we get a very similar sequences. So due to this reason, by using this BLAST program, we can find or identify a species and find the homologous species and the similar species. Okay, so this can be useful when we are working with a DNA sequence from an unknown species. Okay, if we don't know the species of a particular, when we are working with the DNA of a species, so here we can find a homologous species. And second use is the locating domain. When we are working with a protein sequence, you can input it into a BLAST to locate a known domains within a sequence of a interest. So when you practically do the BLAST, so you can understand. So this BLAST program will give you very detailed information about the domains and everything. From there you can compare or know or locate the domains with the sequence of. Okay, third function is or a use of a BLAST is establishing phylogeny. So what is a phylogeny? Phylogeny analysis is nothing but, here we can analyze the most similar species and form a evolutionary tree or a phylogenetic tree. Okay, so using the result received through a BLAST, when the result outcome of a BLAST, when you run the BLAST on the internet or a BLAST program, so the outcome of that result is used to create a phylogenetic tree using a BLAST web page. So phylogeny is based on a BLAST. Okay, so but these along are less reliable. So the phylogenetic trees which are constructed based on a BLAST program are less reliable, though we can create or establish a phylogeny, or we can construct a phylogenetic tree. Okay, DNA mapping. When we are working with a known species and looking to a sequence of a gene at a unknown places or an unknown location, so BLAST can compare a chromosomal position of a sequence. Okay, so in a DNA mapping, so basically the term mapping means it helps to identify the chromosomal particular position of a sequence. Okay, now comparison. When we are working with the genes or a BLAST, we can locate a common genes in a two related species, and this can be used to map a annotation from one organism to another organism. So the basically the whole BLAST major function or a use is a comparison. It is used to compare a protein sequence or a nucleotide sequence in a database. Okay, query sequence against a nucleotide database. So that's it in this video. In the next video we will learn many more things. So thank you, bye bye, like, share and subscribe to my YouTube channel. Thank you.